Welcome, welcome everyone. So we are now very pleased to welcome Dr. Walter Farina for our next plenary speaker. Walter received a PhD from the University of Buenos Aires and obtained a Guggenheim Fellowship to study the behavior and physiology of social insects, focusing in particular on the interaction between communication and cognitive processes in highly social bees at the University of Tübingen in Germany. So he returned to Argentina to spend the subsequent year as a postdoctoral research fellow at the University of Buenos Aires. And then he started as a teaching assistant of biology at the School of Exact and Natural Sciences of the university. And he took up an appointment with the National Scientific and Technical Research Council as an assistant researcher. He became an independent researcher in 2003, combining topics such as behavioral physiology, neurobiology, cognitive and chemical ecology, ecotoxicology, and crop pollination, and using the honeybee as a model study. He is currently a professor at the University of Buenos Aires, a superior investigator at the National Scientific and Technical Research Council of Argentina, and the head of the Social Insect Group study group. Sorry. Welcome, Walter, and thank you for joining us today. You can now start your presentation.
Hi, everybody. Good morning, good afternoon. Thank you so much for the invitation. It's a pleasure for me to participate for this in this virtual meeting. And in this opportunity, I would like to present some recent studies that we performed in our lab, the Social Insect Lab, located at the University of Buenos Aires and in the Institute of Physiology, Molecular Biology, and Neuroscience. In this opportunity, I would like to focus on honeybee cognition topics, particularly in the context of agroecosystems. Our lab, specifically the bee group in the Social Insect Lab, is involved since 30 years in topics related to individual and social behavior, focusing on foraging strategies in social bees, with some connection in neurobiological issues. And in the last 15, 20 years, we began to perform some experiments and approach under natural conditions, focusing on agroecosystems. On one side, our questions are related to cognitive ecology in crops that social bees use for pollinate and obtain fruits and seeds. And on the other side, these agroecosystems also are exposed to many agricultural practices that could be promote and stress to the pollinators. For this, we have to consider, for one side, that the social bees could be a very good pollinator and could improve the forage, the pollination efficiency, and consequently the crop yield of these crops. And on the other side is an excellent sentinel species to determine the degree of stress exposed in particularly in agroecosystems. With this in mind, we have to consider the sharp increase of cultivated area that had promoted a decline in biodiversity in the surrounding of agricultural ecosystems, particularly a very relevant and noticed decline in pollinator biodiversity in these surroundings. So the challenge would be to increment agricultural productivity, but also to take care for environment. Of a consequence of the intensive agricultural practice, there is many examples that had consequence on non-target organisms. Perhaps the best example in this case is the honeybee colony loss that occurred mainly at the beginning of the 21st century, in which, for instance, in the USA, more than 1 million commercial beehives got lost. After this valley in 2007, the population of colonies in USA increased slightly, but if we evaluate the last years of this period, 2016, 2017, focusing on the whole American continent, Europe, and North of Africa, and specifically focusing on winter colony loss, we can observe that the loss of the honeybee colonies in USA is maintaining high, with 40% per winter, in Latin America around 20%, and in Europe around 10%. During this period in USA have defined this abnormal phenomenon as colony collapse disorder that occur when the majority of worker bees in a colony disappear, leaving behind a queen, plenty of food, and only a few nurses bees to maintain caring for the remaining immature bees. This phenomenon seems to be a multi-factor phenomenon in which the environmental stress, the habitat deterioration, air pollution, the intensive use of pesticides, the use of antibiotics, and so on, could cause this mismatch in the social cohesion within these eusocial bees. And for this reason, we think that there is no studies until we decide to become this experiment under realistic conditions, be realistic condition in which we wanted to focus on individual and social behavior and the neurobiological consequence, and also the consequence on the habitat that these societies lives. But to perform these studies, we did an integrative approach. We focusing on individual outputs such as some neurobiological issues, some procedure of behaviors, and also study the interaction between individuals, the global response at the level of the colony. And we have to consider that these colonies should be located or were located in a surrounding in which the human being applied agricultural practice intensively, and for instance, administrate pesticides during the period of planting, seeding, and blooming of the crops. And this implies that when these bees collect resource, also carry pesticide that can carry, that is transport to the hive. This could have consequence at the individual level, but also at the level of the society as a whole. And with this in mind, we perform different studies within the area of biological science. We perform behavioral approaches, evaluating some specific individual and social responses. We evaluated botanical analysis of the food collected by the bees, and also toxicological analysis as product of the food collected. And finally, we perform molecular study focusing on the bee brain involved in foraging in this surrounding. Recently, we published a study in which we tended to correlate the agricultural practice, specifically agricultural intensification, with behavioral abilities of honeybees, and also evaluates the expression of genes in the bee brain that had been candidate to be involved in social responsiveness. We performed this study in the province of Buenos Aires in Argentina in a farm, really a big farm of more than 5,400 hectares. And within this farm, there were located 3 different apiaries. The minimal distance between apiaries was 200 kilometers, and the surroundings of this apiary are exposed in a mix between wild flora and planting of soybean, corn, and so on. During these we worked during 2 consecutive years, and we have the possibility to receive information from the growers of this farm that give us the moment at which they applied pesticides and what mixture of pesticides they had used. During the first year, 2020, the growers applied a mix of herbicides and insecticides, and during the second year, 2021, a mix of herbicides. So our first approach had been to perform honeybee colonies activity measure through the activity at the hive entrance, focusing particularly in the number of incoming pollen foragers, and we can evaluate the free apiaries before and after the pesticide application during the first year and during the second year, and we found no significant differences in terms of activity at the hive entrance. But during a couple of hours at the morning during these periods, we put pollen traps at the hive entrance in order to obtain the pollen collected and obtain a quantitative measure of the pollen collected in terms of weight of pollen. Here we found only significant differences in increase of the pollen collected during the post pesticide application in the second year. And finally, we perform using this pollen collector a qualitative approach in which we did a palynological study that was important in terms to determine what kind of flora the honeybees of this apiary visit. And we pull in 2 different categories, crop flora and wild flora. During the first moment of sample, that is the pre-pesticide application, the bees only collected wild flora, and during the post-application of the pesticide, we observe that the bees collect not only wild flora, but also the crop flora that they were already blooming. So when we evaluate the colony activity and foraging related parameters, we have to say that these 2 moments imply before and after the pesticide application, but also imply before and after the crop blooming. So with the pollen collected at the hive entrance, we perform a chemical approach analysis, a toxicological approach in which we determine the pesticide concentration found in the resource collected, pre and post pesticide application during the first year that the growers offer a mix between herbicide and insecticide. We choose to evaluate only one of the herbicides, glyphosate, and one of the insecticides, chlorantraniliprole. And during the second year that only administered herbicide, we quantified traces of herbicides in pollen collected. We found traces of pesticide not only after the administration of the pesticide, but in some cases before. Moreover, during the first year, we captured some in returning foragers at the hive entrance and evaluate the level of insecticides in terms of traces of this pesticide onto the forager's body. And again, we found traces after, but also before the pesticide administration. So we can say that pesticide applied on the surrounding enter the studied colony and not only throughout the crop blooming. Additionally, we capture forager when returned to the hive, immediately anesthetized, and in a field laboratory, we harnessed the bees in metal tubes in order to evaluate a typical behavioral output in honeybees, that is the proboscis extension responses, in order to evaluate on one side sensory abilities of the captured and harnessed foragers, and on the other side to evaluate cognitive abilities. For the first behavioral assay, we performed a sucrose threshold response assay to evaluate gustatory responsiveness. In this case, we offer to this group of harnessed bees an increasing presentation of sucrose solution and evaluate the positive response by protruding the proboscises. And we found that these two curves are different. The pre and the post administration group show difference, particularly for low sucrose concentration, in which those individuals captured after the pesticide application show a lower response toward the diluted sugared solution. Additionally, we can perform an olfactory proboscis extension response conditioning in which we offer an odor together with a sugar reward and evaluate firstly the learning performance during the acquisition phase, and later we offer only the odor used during the training to evaluate conditioning responses. During the learning acquisition phase, we can observe that in the free apiaries, the bees show a low performance after the administration of the pesticides, and during the evaluation of the conditioning response, also after the administration of the pesticides, the response is significantly lower. In other words, after exposure to pesticides, foraging bees exhibited impaired gustatory sensitivity and cognitive abilities. Additionally, incoming foragers were immediately captured at the hive entrance and freezing, with the idea to analyze in our lab the gene expression of different candidate gene of the bee brain that are related to social responsiveness. For instance, the genes PAL, enolase, and Brahma are defined by previous literature as accelerated evolution in highly eusocial lineages. And also the fourth gene that we evaluate by qPCR is the acetylcholinesterase. All of these are related to connectivity and develop of the central nervous systems. And in all of the cases, we can observe a down regulation of this gene expression during the second moment, that is after the pesticide application. In other words, impaired brain expression level of genes related to neural process linked to social responsiveness. So as a summary of this part, we can quantify two different sample moments. During the first moment, defined as pre-application, no crop blooming are present, and this could be observed in the pollen collected by the foraging bees, because only collected pollen from the wild flora. However, we found pesticide traces in pollen collected and also onto the bodies of the foragers. During the second sample moment, defined as post-application, there is already a crop blooming, and we determined that pollen collected come from wild but also from crop flora. Again, we can detect pesticide traces in pollen collected and onto the body of foragers. And additionally, foragers during this second sample moment show a losing of sense of taste and cognitive ability, and foraging brain expression of genes involved in responsiveness impaired. So the conclusion of this first part is that in agricultural ecosystems, pollinators are exposed to combination of agrochemicals that under laboratory conditions is impossible to evaluate several combination of pesticides together. Also, pollinator behavior impair not only after crop flowering, but also after the pesticide application, and also bee gene expression related to rate of sociability is also impaired. So the relevance of this result, in our opinion, in my opinion, is that this result had been obtained under field realistic conditions, allow us to determine the effective environmental impact of agricultural intensification on pollinators. The second topic, and continue study the agroecosystem. Now we wanted to connect the pollinator with the crop flora, and this is the relevance of pollination in commercial crops. If we consider the last 60 years, it's already well known that the surface, the involved in agricultural practice increased monotonically worldwide. But when we focus on the changes in terms of percentage of the land usage for pollinator-dependent crops, this percentage increased much more higher. Even during the last 60 years, the area devoted to pollinator-dependent crops increased more than 130%. In this scenario, the demand for managed pollinator have increased, and the honeybee Apis mellifera is one of the most required species. This situation forced it to reevaluate strategies to improve honeybee pollination efficiency in agricultural setting. And in order to improve the relationship between honeybees and a crop flower, we wanted to study this relationship in these particular agricultural settings in order to develop some specific procedure that could improve pollination efficiency and consequently, and potentially, of course, the crop yield. In this sense, when we evaluate different level of analysis and focus, for instance, in the inter-individual interaction within the colony, it's very relevant the transfer of food between individuals in honeybee colonies through trophallaxis or month-to-month food exchange. Through this interaction, it's possible that a receiver bee can obtain information related to the richness of the nectar collected, but also can detect or can learn some specific characteristic of the floral type exploited. And this means, in other words, that receiver could establish olfactory memories even it or she doesn't visit the floral patch. And this contact can be fast distributed liquid food among many individuals within the colony, and in other words, the food-related information can be fast propagated. In a context of a pollinator-dependent crops, it's expected that if there is a consensus about some specific floral type, many foragers can visit the crop, and if many individuals collect these results, the foraging efficiency increase, but also the pollination efficiency increase. And this can affect the landscape in terms of, for instance, crop yield of the pollinator-dependent crops. With the idea to improve this relationship between bees and flowers, we aborde different topics within the biological science, from chemical ecology to behavioral science and also pollination ecology. With this in mind, our goal was to promote invasive foraging activity in agricultural setting, focusing, of course, in pollinator-dependent crops, in which bees are considered as essential pollen vectors. Many of these crops offer high-quality fruits, but also could give seeds used for oil, and some of them could consider of low attractive for pollinators. Our goal was to guide the bees to the target crop. This we wanted to think about some procedure that should be as easier as possible, and for in this should be of easy application within the colonies used for pollination service. And we know that the beekeepers, a common practice is to feed with sugared syrup within the colony. With this in mind, we wanted to develop some specific formulation that attract the bees to the target crop. And of course, finally, this formulation that we use it and develop should be environmental friendly for the bee. In fact, we work in different pollinators-dependent crops, from sunflower for hybrid seed production crops, pear and apple crops, and also almond crops. In all of the cases, we develop a simple synthetic mixture that the honeybees confuse with the natural fragrance of the target crop flower, and this stimulation promote an increase in foraging activity biased toward the target crop, improving the crop yield. But today I would like to present a recent study that we published some weeks ago, focusing in a challenge for us because there is some pollinator dependent crops that are very low attractive for bees and even don't offer nectar that is the main attractive resource for a pollinator. This is the case of the kiwi fruit crop. And our first step to analyze this agroecosystem was to identify and quantify floral volatile compounds in kiwi fruits. We know that previous studies published by Twiddle and collaborators performed chemical ecological approaches in which we can observe the profile of the different compounds of the fragrances of the flower of this crop. It's worth to mention that this crop is a dioica species, a female flower, pistillate flower, and a staminate flower, the male flower. The profiles in terms of compounds is very similar, but there is some quantitative differences. And on one side, the staminate flower, the male flower, offer only pollen, while the female flower offer only pseudopollen and no nectar. So with this in mind, we perform in our plots of kiwi fruits extraction of floral volatile compounds in order to study and compare it with previous literature. And we found in two different sites evaluating pistillate and staminate flowers and finding 6 different compounds previously reported in literature that show that this compound elicit honeybee antenna responses. In other words, is other way to say that the peripheral olfactory system of the honeybee can detect at least this compound presence in the fragrances of kiwi fruit flowers. And with this in mind, we wanted to propose a set of potential mimic odor in order to test under controlled conditions, under laboratory conditions. And the behavioral assay that we performed to evaluate the different candidates for mimic odor is to perform an olfactory proboscis extension response that during the acquisition phase, we offer the potential mixture together with odor, and at the end of the learning phase, we expose the bees to the extract of the fragrance of the female and the male flower in order to evaluate odor generalization. Here is the result that we obtained. We test 6 candidate odor and mixture as potential mimic odor to use it in kiwi fruit crops, and we found that all the curves really are seemingly good. The bees learn all the synthetic mixture, but one of them, the KM, present the highest response in terms of generalization will present the extract of pistillate flowers and also present one of the 3 maximal responses when offer the extract of staminate flowers. So we decide to use, because the KM mimic odor show the highest level of generalization to the natural flora sense to perform with this mimic odor experiment in the field. So we visit a kiwi fruit crop at the province of Buenos Aires, Argentina, during 3 consecutive years, during, of course, the blooming period, and stimulate the colonies, the beehives used for pollination service with 2 different treatments, one group with unscented sugar solution and the second with the scented sugar solution. When we evaluate the activity at the hive entrance in terms of incoming bees and in terms of incoming bees with pollen in general terms, we found no differences between the conditioned and the control groups. But when we use it pollen trap during the couple of hours in the morning and could discriminate the pollen collected onto the kiwi fruit flowers that we define it as white pollen load, compared with the rest of the pollen collected, we observe significant differences between the 2 treatments. The conditioned colonies showed an enhanced kiwi fruit pollen collection. Even more, we perform an additional pilot trial in a second plot at the province of Buenos Aires, in which we stimulate colonies with these 2 treatments, and branches of this crop closer to one colony treatment or the other was marked, and during the period in which they began to establish the fruit set period, we quantify the number of fruit per flowers, obtaining that the fruit set of kiwi fruit plant in the surrounding of treated colony resulted higher than the control plants. So, as a summary of this, we can say that we have a candidate of mimic odor to use in this particularly crop. So, as a summary of our targeted pollination, considering all the crops that we study, the first step would be to visit the pollinator-dependent crop during the blooming period, obtain extract of floral volatile compounds in order to analyze chemically this extract, and this will be useful to define a set of potential mimic odor that should be as simple as possible through a laboratory experiment, in this case the olfactory condition, and those candidates that show the most degree of generalization compared with the extract of the natural fragrances had been used in field experiments in which we scented the food that we offer to the colonies used for the pollination service, assuming that this establishes specific memories in many individuals within the colonies, and some of them, when began to perform foraging activity, show a biased preferences toward the crop, and this promote an increased crop yield. But additionally to the complexity of the kiwi fruit crops that show that had been defined as a nectarless dioicous crop, additionally show that this crop present a very brief blooming period between 10 and 14 days, which is very difficult to use a common practice define it as pollination service. For this reason, we wanted to improve this mimic odor that we developed for kiwi fruit crop, adding some additionally non-nectar sugar compound that we know that could be used as tonifier in pollinator. This is the case of the alkaloid caffeine that could offer by different genera such as coffea and citrus, not only in nectar but also in pollen, and that is defined as a tonifier, and also many floral resources offer amino acids, and one of them is the arginine, and arginine is a precursor or nitric oxide that in the central nervous system is very important to stimulate the biochemical cascade to promote protein synthesis. In other words, to stimulate a process involved in long-term memories. We know that caffeine had a positive effect in the learning acquisition in pollinators, particularly in honeybees, and promote long-term memories. There is also information that arginine could improve short-term memories, but there is only a few information that what happened when we combine both of these non-sugar compound that could be collaborate on different bias in the processes or learning processes. We already studied under laboratory condition in which if we perform an olfactory condition in which the reward is sugar solution and containing an element of this non-nectar compound or combine it both, we obtain that when we combine caffeine and arginine in a specific concentration, this improves cognitive abilities, particularly a high proportion of experimental subjects show long-term memories responses. And we repeat this experiment under field condition, specifically in pollinator-dependent crops, such as sunflower for hybrid seed production that mainly offer nectar, but also pollen. And again, we observe that those colonies treated with a combination of both non-sugar compound show an enhanced pollination activity, and even in the surrounding of the treated colonies, an increased crop yield. But what happened in a so particularly crop such as a nectarless dioicous crop with a very brief blooming period? This is the case of the kiwi fruit. And we use here a kiwi fruit plot, and we located 4 groups of 15 beehives treated with all the 4 groups with this KM empty sugar solution, but the control group received only KM empty sugar solution, and the rest with or one of the 2 non-sugar compounds, caffeine or arginine, or the mix of both. Later, we focus only on the pollen collected by these colonies by using pollen traps. And when we evaluate the collection of kiwi fruit pollen exclusively, we found significant differences between treatments, but particularly when we focus on the number of kiwi fruit pollen load and the proportion of kiwi fruit pollen load, we observe that the colonies treated with the mimic odor plus caffeine alone show a lower response in terms to collect kiwi fruit pollen. And this is surprising because we expect that caffeine improve foraging activity, but all the report previously published focused on food and feeding places that offer sugar solution or even under natural condition bees that collect nectars as resource. And even more, when we evaluate quantitative the weight of the pollen load collected per individual, we found significant differences in which only those colonies treated with the mixture show an increase in size, a heavier pollen loads compared with the control, and the rest of the treatment are slightly lower in terms of weight compared to the control. Additionally, we studied the amount of Tory pollen in beehives and we compared the area occupied by the pollen collected before and after the blooming period, and only those colonies exposed to the KM-scented sugar solution plus the mixture of caffeine and arginine show a significantly increase compared with the control. So we can say that this treatment promote collected heavier kiwi fruit pollen loads and showed an increased pollen storage area in the beehives used in the pollination service. So as a summary of this talk, we can say that behavioral tests in the lab and in the field allow us to understand how social bees use floral information to make decisions in complex appetitive environment. Additionally, we have developed a platform of specific mimic odors for different pollinator-dependent crops. We work in sunflower, pear, apple, almond, blueberry, kiwi fruit, and so on that improve the foraging activity of treated beehives. Thirdly, combining non-sugar compounds enhance the action of the mimic odor, promoting colonies with higher foraging activity in the crop, even in a nectarless crop. So if we consider this relationship between honeybees and agroecosystem, and we can consider the both sides of the same coin, and if we wanted to avoid or minimize the activities of this pollinator in a disturbed environment such as agroecosystem, we think that this targeted pollination procedure could promote to reduce the number of colonies used for pollination service and or to reduce the time spent in the crop, minimizing the health risk of the beehive colonies. So I would like to acknowledge all the members of our group that during these more than 20 years had been involved or are now involved in this procedure. Thank you very much, all of these nice people that collaborate and participate in this project. And thank you so much for your attention. I am open to your question. Thank you.
Thank you, Walter, for this very interesting talk. So for people who are listening right now, it's time for question. You are free to ask questions in YouTube, and I will read them to Walter. We already have a few questions. So one from Hilton. Multi-species crop systems among a matrix of wild vegetation area would not help better the bees to increase crop productivity? Isn't this a way to align nature conservation and human productivity?
Yes, in some way the use of this procedure have to consider not only the benefit of a grower in turn to increase productivity, but also to care the pollinators. And we think that if we the bees are exposed in a disturbed environment, such as an agroecosystem, of course, the best is that this agroecosystem are around a wild flora, which improve the healthy condition of the beehives. But with this procedure, it's possible to reduce the exposition of these pollinators in this agroecosystem.
I see. We have another question from Catherine. Thank you for this great talk. I think it is interesting that you are able to increase foraging by exposing the hive with the KM or odor. Does it have an impact on other plants? For example, by shifting the focus on kiwi fruit, do we see a reduction in pollinating wild plants?
Yes, in some way, with this procedure, we stimulate with sugar solution that many individuals become to collect resources in the surrounding of the beehive. But we have to know that the feeding places offer only pollen. So in this case, we discard the great foraging group that collect nectar, and we consider only those groups that collect pollen. And we focus this activity only through a brief period in which it's possible to establish long-term memory, activate to this resource, and immediately when this resource is already empty, it's possible to initiate or reactivate activity in wild or alternative resources.
Perfect. I also have a question. So pesticide decrease cognitive and social abilities, and I was wondering, can it come back if bees are not more exposed, or is the loss definitive for the bees?
For this reason, I said that there is the two the two sides of the same coin, because really the agricultural practice and also the agricultural practice performed in pollinator-dependent crops use many pesticides, and it's expected that growers keep in mind the relevance that pollinators should avoid these poisons material. For instance, perform the application during the night, or if there is no particular pathology on the flowers, avoid to use pesticides. But the idea would be also, as I said at the end of the talk, to reduce the time spent of pollinator within this agroecosystem, because are very exposed to pesticide in detriment of many related neurobiological aspects. For this reason, I think that is important, these considerations.
Thanks for the answer. So Catherine said, thank you for your answer before. And we have another question from AB. A great talk. Are you planning to integrate some experiments on the dance communication like you had done before with your current work on pollen collection?
Most that is much more easy to work in systems that offer mainly nectar, and there is only a few crops that offer pollen. The kiwi fruits is particularly relevant from term of productivity, nutrient for, and also commercially, but nowadays we are involved in some of these type of crops only.
I also have another question. So I'm not a bee person, so I sorry if it's a idiot question, but So for me, it was amazing that we can train bees to go to crops with the mimic odors. I didn't even know about this. But in this case, do you train just a few number of bees and then you count on them to show the path to the others, or do you train like all of them at the same time from one hive?
Yes, but that depends of the blooming period of the crop study, but mainly should be at the beginning of the blooming period when the pollination service companies locate the colony in the surrounding, and with 15-20% of blooming in the crop, is the best moment to stimulate one time with scented sugar solution. This should be enough for, I, I estimate, around 10 days. But for instance, some crops, such as blueberries, in which the blooming period is around 30 days or
Yeah,
more, we need much more stimulation. For instance, twice or three applications in order to maintain these
that's right.
olfactory memories working.
Yeah, makes sense. um, so this is the end of the question period, I think. I think we will have much more question in Discord if you go on Discord, because I know that we are a lot of bee lovers on ABL. So please go, you will have much more question. And you can now leave the session. Thank you again, Walter, for this like very interesting presentation. And we will go now for a little break. If I can see my mic. Okay, it's here. So see you in a few minutes for the first talk of the session. And it's just time for the little break now. Thank you again, Walter.